Antigen. This one is the new topic in the immunology. Dr. Ram Suthar, Department of Biotechnology, PSH HDA Kadi. Antigen. Antigen is defined a foreign substance that is satisfied with the two distinct immunologically or properties. The number one is immunogenicity and number two antigenicity. Proteins, carbohydrates, lipids, nucleic acids are the components of the antigens. Antigens are the large molecules with 10,000 Dalton. Immunogenicity. It is the ability to an antigen to induce immune response in body. So the both humoral and cell mediated, it is the able to induce immune response. The B cell are attached with the antigens that affected B cells. This one is known as the plasma cells and the memory B cells. In the T cells, they have attached to the antigens and the affected T cells, that is known as the helper T cells and cytokine T cells with the memory T cell. Substance that satisfy this property, immunogenicity, and more appropriate called the immunogens rather than the antigen, because these substances can be stimulate the immune response, so that is known as the immunogen. Ability to an antigen to combine specifically with the final product antibodies or the T cell surface receptor. All molecules having immunogenicity property also show antigenicity, but the reverse is not true. For example, heptans. The heptans is the molecules, that is a small molecules. They have antigenicity but not immunogenicity, because the heptan cannot be stimulate epitopes, the smallest unit of the antigenicity. If we define that is a small area of the antigens with the 4 to 5 amino acids or the monosaccharides residues that is capable to sensitize T cell or the B cell and reacting with the specific site of the T cell receptor or an antibody. Specific site of an antibody that react with the corresponding epitopes of an antigen is called as the paratopes. So the paratope is present on the antibody or the epitopes is present on the antigen. So the antigens have a specific things or specific some molecules or residues that is known as the epitopes, and that epitope is attached to the antibody with the specific region, and that region is a paratope, and that paratope region is on the variable heavy chain or the variable light chain of the antibody. These are the two types, subsequently and the linear epitopes. So the simple or the single linear sequence of the few amino acid residues are known as the linear epitopes, and the non-subsequence epitopes is flexible region to complex antigen having the tertiary structures. They form by bringing together the surface residue from the different site of the peptide chain during its folding into the tertiary structure. T cell recognize sequentially epitopes, while the B cell bind to the conformational epitopes. Heptans. The heptans are the lower molecule weighted